All right, ladies and gentlemen, welcome back. We are going to continue wrapping up talking about your targeted defenses or your third line of defense, your adaptive immune system, by getting into exactly what happens to those lymphocytes, those B and those T cells, once they find a corresponding antigen. What happens to them? So let's take it a step back. Before that antigen is even encountered, these lymphocytes are in what we talk about as a naive state, meaning that they've gone through their stem cell to differentiation. They are either in your bone marrow, they're in your thymus, they are already predetermined to be B or T cells, and then they go out into your lymph nodes, go out into your tissue fluid, go out into your blood, and they're just kind of circulating at this point. Right now, although they are B cells or T cells, they haven't found their corresponding antigen yet, and so they're not really doing much of anything other than just floating around, waiting for that connection to be made. So on these naive lymphocytes, they have recognition sites specifically shaped to a potential antigen that exists. And we'll get into a little later about the genetics of how these different receptors get created. But basically, you have hundreds of different types of shapes sitting at the ends of these receptors in 100 different types of B and T cells that are just floating around your body, waiting to find that recognition. So these guys oftentimes tend to hang out in your peripheral lymph organs once they have been released from your bone marrow or your thymus. They tend to hang out in your lymph nodes more often than not. Um, this is where that site of antigen connection concentration really ends up starting when things like macrophages or some of those cells from those innate immune responses bring those antigens from the initial site of infection to these lymph nodes for this targeted defense to actually start happening. So once we get a situation where those naive lymphocytes make a shape connection to their corresponding antigen, we'll then go through this process called clonal selection. So here's this pool of mature but naive lymphocytes, right? So they're floating around in your lymph nodes, in other tissue fluids. They've got all different options in terms of antigen recognition sites. But they're in relatively low numbers. They're just a handful of each of these floating around at that point in time. Once that antigen gets presented to these naive lymphocytes, we then decide, okay, this is the one that has the corresponding shape to the antigen that's currently present, and so this is the one that I'm going to now go through clonal selection and go through proliferation. Basically, I'm going to make lots and lots and lots of extra copies of this particular lymphocyte because I've now recognized that that's the connection, that's the antigen that fits. And so that's the one that I need to help ramp up this targeted immune response. So the phases of lymphocyte activation take time, right? So when I get through, here's my naive B cell, for example, and that antigen now gets connected to those receptors. So this is the one that I'm now going to decide goes through that proliferation, and I'm going to make lots and lots and lots of them, right? So I go through that clonal selection process. And then I need to get into my differentiation piece where I'm going to take those B cells and they're going to become antibody secreting cells, they're going to become memory cells, so on and so forth. This is the actual fighting of the infection. So up to this point of proliferation, you may be showing symptoms of the infection because your body hasn't had the chance to ramp up that immune response enough to actually start fighting off the infection. The whole idea here is once that proliferation occurs, I need to be able to keep pace with that replicating microbe. And although that phase is relatively quick, right? One B cell can become 4,000 antibody secreting cells, which equates to about 10 to the 12th antibodies produced per hour, but that's only once I've gotten past this point. So it's going to take some time from here to here, and that time will depend on whether or not your body has come across these antigens, this pathogen before. If it's the first time you're coming across this, we'll talk about your primary immune response, and this may take days before we get from here to here and we're actually fighting off the infection. Where if it's a secondary infection, second time you've come across it, a secondary immune response, this will go much quicker. And so you'll be able to fight off that infection perhaps in a quick enough period of time that you don't even know that you've been infected because you're not ever building up enough bacterial load or viral load or parasite load, whatever the pathogen is, to actually feel the effects of it being there because your immune system just wiped it out quick enough. But that's only if you've come across that before. So here's kind of what I'm getting at, right? So the need for that proliferation, that need for that differentiation has a delay, especially if it's the first time you've ever encountered it. And so it's typically anywhere from 4 to 7 days in somebody who has a truly effective adaptive immune system, if this is the first time you've come across it. So here are those naive B and T cells hanging out. You've got that initial recognition that will happen within the first few days, but then it's going to take 7 days, 4 days, 7 days before you get enough antibody production going on from that differentiation to actually start targeting, tagging the infection, and then for your T cells to take care of the infection itself, go through that humoral and cell-mediated immunity, destroy those cells before we're finally seeing you return to homeostasis, aka the infection has been cleared. Any of those excess cells then that you created in this process that are no longer needed because the infection is cleared will go through apoptosis, which is essentially programmed cell death. We're going to get rid of those excess cells so that you're not just hanging on to insane amounts of B and T cells all the time. That's not necessary. So to make efficiency return, we'll get rid of them, but then you will have some surviving cells that hang out that are your memory cells, the cells that kind of remember that specific antigen, that specific pathogen. Again, so that if we come across that next time, the piece over here happens much more quickly because these guys already exist. And so we've reached this point of differentiation, clonal expansion, the actual elimination of the antigens in a much shorter time period. So once we get that activation that occurs, we then start looking at the effector lymphocytes, your effector B and T cells. And these are those activated lymphocytes that are now capable of performing the functions required to actually eliminate the pathogen. So when we talk about effector T cells, these are going to include your helper T cells, which are going to be mainly there to create cytokines and do cytokine secretion to sort of just pull together and activate all the other innate and adaptive pieces of your immune system to say, yep, there really is a problem here. We want to ramp up that immune response and take care of it. And then you've also got other effector T cells that will go through and actually start killing infected cells. Your B lymphocytes really go through the differentiation to become those plasma cells, and those plasma cells are the ones that do the antibody secreting, so that they can go those antibodies can then go tag antigens that they find in your body, so that the T cells then can come in and destroy those pathogens. These effector cells, though, are relatively short-lived. They act immediately once they've been created. These are going to be the ones, though, that go through that apoptosis, and we get rid of them once the infection is cleared. These guys aren't going to hang out in your body for long periods of time. A couple of things to keep in mind while we're talking here about these targeted defenses. B cells are monitoring your extracellular fluid. All right, these are the ones where those antibodies are getting created, and it is in that extracellular fluid, whether it's your blood, your lymph tissue, your tissue fluids, where those antibodies will ultimately attach. Again, remember that those antibodies that those B cells are producing, those plasma cells are producing, aren't going to actually directly destroy the pathogen or the infected cell. Instead, they're going to inactivate that pathogen, inactivate that virus, inactivate that cell or that molecule that's causing you disruption until something else comes in to destroy it. And most of the time, once that antigen antibody complex is formed, it is then the T cells that come in to take care of that. Some of your T cells will be your helper T cells, so they'll secrete those cytokines to go ahead and say, hey, let's ramp up this problem, or let's ramp up this response, I should say, to eliminate this problem. That will also further activate B cells to continue pumping out those antibodies. The actual clonal selection process is the same for both your B and your T cells. They need that antigen recognition to connect to the same shaped receptor that exists on their cells. So you're clonally selecting both for B cells and T cells at the same time. The only difference is, remember that your T cells have that MHC. They do that dual recognition. So they not only recognize the antigen, but then they do that double check just to make sure that this is in fact a problem and it's not trying to attack your own cells unless it's necessary. And so there's a lot of cooperation happening between those B and T cells. So although we're going to kind of talk piecemeal here, remember all of this is going on more or less at once. So I mentioned to you that the antibody production is specifically controlled by your genetics. So you have immunoglobulin genes that encode for one chain of the B cell receptors. So here's your B cells. All right, we're basically looking at genetic information within your B cells, and those B cells then will produce these antigen receptors on their cell membrane. And you've got a set of genes here that control this production, right? So this piece right here. And the way in which we can create all these different varieties or different shapes in this antigen recognition site is to be able to go through and look at expressing that set of genetic information in different combinations. So I have all of these genes that are controlling for that little arm of that receptor, but I don't necessarily express all of it at once. So if I take this piece here, right, I've got these individual genes, okay? When we go through transcription, we're going to make RNA copies of this. We get to a differentiated B cell. Now it only contains this functional set of genes for this particular antibody. I go through and create that pre-messenger RNA, right? We have to do splicing and removing excess information that doesn't actually code for proteins. Hopefully maybe exons and introns sound familiar to you. So we go through that processing, and then we get to the point of translation where I'm actually building this. So that differentiation piece from here to here determines what set of genes I'm actually going to express once I get down in here and produce this. So every cell in your body, just sort of a side note here, contains all of the genetic information. It's identical. If I take a cell out of your skin versus a cell out of your brain versus a blood cell, they all contain the entirety of your genome, all 46 chromosomes worth of information. And yet, in order for your white blood cells to do one job, your red blood cells to do another, the muscle in your heart to be different from the muscle in your arm, to the neuron in your brain versus a sensory neuron in the tip of your finger, all of this comes down to only expressing certain parts of that genetic information. So when we look at your B cells, they are able to take that collective set of genes that are necessary to create those receptor sites and only express a subset of them. So hopefully that makes sense. So rather than this all exists in that B cell, but once it goes through that differentiation process, we're now only expressing this part. I get to another B cell, and now I may express a different subset of that set of genes. So rather than it being 39V and 5J, I may be 39V and 3J, so on and so forth. So I can take different combinations here and be able to create different recognition sites. Think of it like the alphabet. If I can take all those different letters and put them together in 2 or 3 word combinations, I can create this huge language, and that's exactly what your cells are doing here. So this is just a diagram to kind of walk you through this again. So here's the antigen recognition, right? Activating the innate immune cells, taking those naive cells, differentiating them, and what are they doing in terms of secreting antibodies for your humoral immunity versus your cell-mediated over here. So it's just kind of another way to summarize what we've been talking about. All right, so on the back end, once your body has taken care of that infection, most of those effector cells will go through apoptosis and get destroyed. We're not going to carry around all of these crazy amounts of white blood cells all the time. That would be taxing on your body. So we destroy them. That being said, a subset of those will differentiate then into these memory lymphocytes, and you have memory B cells as well as memory T cells. These are the ones that are long-lived. These are the ones that are hanging out in your body just waiting to find that same antigen again. But they're functionally silent. They're just sitting there waiting. They're not actually doing any sort of immune response. They're just monitoring to see if they come across that again. And these memory lymphocytes are capable of going through that differentiation and proliferation phase much, much quicker to make those effector lymphocytes the second time or the third time or the fourth time that you come in contact with that same antigen, that same pathogen. And these are the cells that really mount that secondary immune response that we're going to talk about, which is much quicker and much more effective, meaning that a lot of times you don't even feel symptoms when you come across that pathogen again, at least for some period of time. The big question becomes, how long do these memory cells stick around? For some diseases, it seems like they stick around for life. For others, they sort of teeter out after a certain period of time. And at this point, we don't fully understand why you keep the memory cells for some and not for others.